Hi everyone, I am Leonard Dervishi and I'll be presenting the paper with the topic Privacy-Preserving Identification of Population Stratification for Collaborative Genomic Research. Below are the rest of the authors. Initially, I would like to give a brief outline of today's presentation. I'll go into the introduction, provide some background information after that. Afterwards, I'll provide the system and threat models along with the proposed framework. I'll dive into the privacy analysis, and finally, I'll provide the results and conclude the talk. Collaborative research has been more and more important in various areas in computer science, but especially for genomic research, since it usually produces more accurate outcomes and powerful statistics. The main privacy implications for genomic data include phenotype, family memberships, disease conditions, etc. And before performing the collaborative research, we also need to ensure the data quality. We can do that using the quality control steps in genomic studies and also in GWAS, which stands for Genome-Wide Association Studies. The goal of GWAS is to find the genetic variants that are associated with a certain disease or phenotype. And one of the most important steps of quality control is population stratification. As a concept, it stands for genetic differences that are unrelated to the phenotype, but due to ancestry. And what we need to do is that we need to remove this bias. Principal component analysis is a popular technique which identifies the patterns within the data. And as we can see on the right, we're only using two principal components for that PCA plot. The genomic researchers, they've been using the PCA outcome to assign individuals to populations. And we also need to make sure that the study participants are selected from a relatively homogeneous population. The goal of this paper is to obtain an accurate PCA outcome of the federated datasets and also preserve the privacy of the samples. So the main challenge that we have tried to solve is identifying populations across distributed genomic datasets in a privacy-preserving manner. I'll want to provide some background information first regarding the local differential privacy, and after that I will go also on the PCA training. Differential privacy is about sharing statistics about the dataset while preserving the privacy of the individuals. It can be done using statistical noise to the data, either to the input or to the output, and local differential privacy, it usually adds data to the input. And epsilon LDP could be achieved by different methods, such as randomized response, but in our paper we've been using addition of noise from a Laplacian distribution. After that, we also have some background information on PCA training. We know that principal components are computed from the original variables and capture the most important patterns in the data. And the top principal components are usually used to transform the data. They are adjustable in the sense that we could use two of the main principal components or more than that. For our paper, we've been mainly using two, and I'll dive into the details of that one later. PCA training usually utilizes SVD to extract the components of it, and the PCA outcome for new data points is computed by taking the dot product of these fitted principal components with the new data points. For the system model, we consider two main parties. We have the researchers and the server. The researchers generate and provide the metadata to the server, and the server tries to label each individual to their respective population. In the threat model, we consider honest researchers who provide accurate metadata and do not act malicious. But on the other side, we assume honest but curious server, which means that the server follows the protocol, but may also try to infer some sensitive information from the data. The main privacy attacks used in genomic research are the following. Attribute inference attack, where the adversary tries to infer sensitive attributes using the available auxiliary information. We have de-anonymization attacks, where the adversary tries to match the identity of a victim with one of the participants in a genomic dataset. And we also have membership inference attack, where the adversary has access to the genomic data of a victim and tries to infer whether that victim is a part of a genomic dataset or not. The most relevant attack to our scenario is membership inference attack. Now I'll dive into the details of the proposed framework, which has 3 steps. In the first step, the server trains a PCA model using a publicly available genomic dataset, which contains individuals of various populations. Next, the trained PCA model is sent to each of the researchers. In step 2, the researchers transform their local datasets using the model that was sent to them. As a result, each researcher obtains the projection of individuals in their dataset with respect to the trained PCA model. In order to achieve local differential privacy, and hence to protect the privacy of the records in their local datasets, researchers add Laplacian noise to each of the samples. In the last step, the researchers send some metadata, which contains the dimensionality reduced data points, or we can also call them PCA coordinates, and the hashed IDs of each sample to the server. The server then calculates the coordinates for all the users coming from multiple researchers. It combines those PCA results and identifies the population substructure and the population clusters. Finally, it sends back to each researcher the hashed user IDs and the label of the population cluster they belong to. For the privacy analysis, we have the adversary who operates at the server side and has access to the following: the PCA output for the whole collaborative study. It also has access to the trained PCA model and the genome of the victim. Since the adversary has access to the genome of the victim, it can also generate the coordinates of that victim in the PCA output. Now, the privacy risk in this case is the server. It's trying to match a victim's coordinates with any of the samples that are part of that specific dataset. Why is this dangerous? It is because the dataset may contain a sensitive attribute or phenotype such as the HIV status. In order to quantify the privacy analysis, we use the membership inference power analysis, which is based on the Euclidean distance between the PCA outputs. What we do is that we use a controlled set of individuals that are not in the dataset and compute the Euclidean distance between their PCA outputs and those of individuals in the dataset. The minimum Euclidean distance is identified and is used to determine a threshold. Then a set of individuals in the dataset are analyzed, and their minimum Euclidean distances are compared to the threshold to measure the membership inference power. Furthermore, we also compare privacy risk of the framework with a baseline, which is sharing of aggregate statistics and is allowed by many institutions, including the NIH. In order to evaluate our framework, we use the following 2 datasets. The first one is OpenSNP dataset, which has 120 samples and 9,000 SNPs. And the second one is 1,000 genomes phase 3 datasets, which contains 600 samples and 37,000 SNPs. The metrics that we use are precision, recall, and power. For the precision, we define it as the ratio of the correctly classified individuals to a cluster over the total number of classified individuals to the same cluster, while the recall is over the total number of the individuals belonging to that cluster. And for the power, we use the membership inference attack that we explained in the privacy analysis. Initially, we evaluate the impact of varying the number of populations in the server's dataset for training the PCA model. These 4 experiments are designed by modifying the content of the dataset. The centralized PCA combines all datasets and cluster samples using k-means of the PCA output, and the resulting clusters initially serve as a benchmark. Then the proposed framework's accuracy is assessed by changing the content of DS, and it achieves accuracies of 0.92, 0.97, and 1 for different population combinations, as shown in parts A, B, and C, where in A there is only 1 population training the dataset, in B there are 2, and in C there are 3. Furthermore, we also have in D, where all 5 populations are included in training the dataset, and the accuracy achieves this one. What we notice is that the performance increases as the number of populations in DS grows, and even with an incomplete representation of locally observed populations, high accuracy is obtained. Next, we evaluate the trade-off between utility and privacy. We first consider the scenario where the datasets consist of one population each, which are different from each other. And we notice that as the epsilon value increases, the level of noise decreases, and that results in both higher utility values, both precision and recall, and an increased power for membership inference. You can see that for epsilon 3 and k is equal to 3, precision and recall are close to 1, and the power is 0.2. Small clarification, k here is for the k-means algorithms, which is the number of clusters. We notice that an increasing k enhances utility in terms of precision and recall, and the number of clusters aligns with the optimal value. So the frameworks perform much better when that value is achieved. We also consider the scenario where both datasets DA and DB contain samples from two different populations. Such a scenario is more realistic, and we observe similar results in terms of utility, but the power is almost halved. This is due to the fact that even though the attacker can determine the population of a target victim, it is hard for him to determine to which dataset it actually belongs. Next, we evaluate the effect of providing different number of principal components, which also corresponds to the number of dimensions of the PCA output. What we notice is that the results for different number of dimensions were very similar in terms of utility and membership inference power. So considering that usage of a higher value of dimensions would introduce computation overhead, the researchers are inclined to use only the first two principal components while using our framework. Furthermore, we conducted a comparison between the privacy risk of our proposed framework, which involves sharing PCA output, and the risk associated with sharing GIVA statistics, which is referred as the baseline risk. In our experiments, even with just 10 GIVA statistics obtained from a dataset of 120 samples, the baseline risk has a power greater than 0.44 for membership inference. We also notice that this power increases as the number of return statistics increases. And since researchers typically share tens of statistics as a research outcome in GIVAs, when we compare the achieved power with the power observed in our experiments, we find that the power of Euclidean distance on the PCA output does not exceed 0.4 in all of our conducted experiments. Even when considering a dataset with a similar number of samples, NT, which is shown in figure C, the pink line, the power of our framework is still under 0.44. Based on these results, we conclude that the privacy risk associated with our proposed scheme is lower than the risk posed by sharing GIVA statistics. As a final result, we demonstrate the running time of our framework. We conducted a few experiments and inspected the change in the running time for different scenarios. We show the running time at the server side, which consists of the time needed to train the model and apply k-means algorithm on the received metadata. The other column shows the researcher side, which includes reducing the dimensionality of the data and the addition of noise, and the total running time. Out of all the operations performed, the training of the PCA model has the biggest running time, and we also found out that the running time increases linearly as the sample size per dataset grows. Additionally, when researchers' datasets contain samples from multiple populations, the running time on their side doubles. We observed that for a dataset which has over 37,000 SNPs and 600 samples, the framework completes its execution just for a couple of seconds. Therefore, we conclude that the proposed framework is both practical and efficient. While population ancestry is often viewed as a discrete notion, it can also be a complex and continuous concept depending on the context. For example, certain individuals may have mixed ancestries, which makes it even harder to accurately classify them into a specific population cluster. This is an issue inherent to PCA and cannot be addressed by our framework as well. Nevertheless, there are a few major population clusters which are widely accepted by the scientific community, and we also notice that most of the samples fall into one of these clusters, and we classify the samples that lie between the clusters into the closest one. A minor limitation of our framework is that a single individual belonging to a population different from all the other ones is at more membership inference risk. A countermeasure to such a scenario would be to greatly increase the amount of added noise, but that would degrade the utility of the proposed scheme. However, such a scenario is very rare, considering 100 or maybe even 1,000 of samples that are part of that study. Summary of our work, we have proposed a novel and effective privacy-preserving framework, which mainly partitions populations in collaborative studies. Via experiments, we just showed that the proposed framework identifies with high accuracy, precision, and recall the genetic differences among collaborator datasets, while preserving the privacy of the research participants. We also showed that our proposed framework keeps the privacy risk below the baseline risk of sharing summary statistics. Hopefully, in the future, this work will enable researchers to conduct collaborative research with high-quality data while ensuring that the privacy of the research participants is preserved. Please feel free to contact us if you have any questions, and thank you for listening.